Alright, my name is Kaylee Yeager, and the date today is October 20th, 2020. So today we'll be talking about the role of the pancreas and liver in regulating digestion. We'll begin this discussion by talking about the anatomy of the liver, and it's important to note that the liver is the largest gland in the human body, and this is because it has many important functions outside of just regulating digestion. There are two main lobes. This is the left lobe over here, and the right lobe is right here, and they're separated by what's called the falciform ligament. What we're most concerned about in regulating digestion are these three structures, and they all enter or leave what's called the porta hepatis. We'll start by talking about the portal vein, which is the structure in blue. And the portal vein delivers partially deoxygenated blood from the GI tract, pancreas, and spleen to the liver, and contains many nutrients and toxins. One of these nutrients is bile salts, which are returned to the liver via the portal vein, and we'll talk about this later in the lecture. It's actually not a true vein because it delivers blood to the capillary beds in the liver and not straight to the heart. The second structure is the hepatic artery, and this delivers oxygenated blood straight from the heart to the liver. And then the third one that we'll be discussing is the hepatic duct, or the bile duct, and it drains bile from the liver and transports secretions from the liver to the small intestine. It's important to note that blood and bile flow in opposite directions. And then finally, hepatocytes are liver cells that secrete bile, and these are important because we'll talk about them later in this next slide. So the main secretion of the liver is called bile, and there are three main components of bile. The first one is bile salts, and these facilitate enzymatic fat digestion. There are bile pigments, which are waste products from the destruction of aged or abnormal red blood cells. For example, there's bilirubin, and there's also its oxidized form, biliverdin. And kind of just like an off-topic note, bilirubin is orange or yellow in color, and its oxidized form is biliverdin, which is green. And when they mix with intestinal contents, they give the brown color to feces. Also, high levels of bilirubin can indicate many diseases, but we won't get into that here. Bile salts, and finally there's cholesterol. Bile salts is what we're going to be most focused on here because they act as emulsifying agents by separating lipid clumps and increasing their surface area. So this little diagram over here, we can see that bile salts enter, and then these coat the large droplets that come from the stomach, and then they create what's called an emulsion. And this is very important because it makes these lipid clumps water soluble. And eventually they get broken down into smaller molecules or clumps called micelles. And this happens with the help of two main enzymes. The first one is pancreatic lipase, and then there's also cholipase, which break down the fats into monoglycerides and two fatty acids. And that's what's stored in the micelles. So lipase is the enzyme that removes two fatty acids from the triglyceride molecule, and cholipase displaces the bile salts and allows the lipase to access the fats inside of the bile salt coating. And then third here, you can see that the monoglycerides and the fatty acids move out of the micelles and enter cells of the small intestine via diffusion. And then you can also see that the bile salts are recycled after they reach the ileum. They're converted to bile acids, and they are brought back to the liver via the portal vein. And eventually the hepatocytes excrete bile salts into the newly formed bile, and this is important because we want to continue using these bile salts without having to produce more. Next, we'll be talking about the anatomy of the pancreas. So as you can see in this diagram over here, the pancreas is very closely associated with the small intestine and also the gallbladder. And you can also see here this small line is the pancreatic duct, and it enters the duodenum, or a portion of the small intestine. And there are three main cells in the pancreas. The first one is acinar cells, and you can see those here in this cross section, and these secrete digestive enzymes. Then there's also these lightly stained regions called the islets, and they secrete hormones. And finally, there's these duct cells, and they secrete bicarbonate solution. And there are a mix of endocrine and exocrine functions, which are important in regulating digestion. So we'll start by talking about the exocrine pancreas secretions. And the first major secretion is digestive enzymes, and they are secreted in their inactive form, and they're eventually activated in the duodenum. So like I said previously, they are secreted by the acinar cells, and they are activated in the presence of enteropeptidase. And in this diagram over here, you can see that the pancreatic secretions are the inactive zymogens and trypsinogen. So that what the enteropeptidase does is it cleaves the trypsinogen into trypsin, and the enteropeptidase is located in the brush border of the small intestine. And this brush border is made of microvilli that increases the surface area of the apical membrane, and it increases the total area for absorption as well. And eventually, trypsin activates these inactive zymogens and creates them so they're in their active forms. And these digestive enzymes are signaled by the release of a few different things. So the first thing is small intestine distension. There's the presence of food in the intestine, neural signals, and cholecystokinin. And this is a GI hormone that stimulates the digestion of fats and proteins. And there are three very important digestive enzymes that I'll discuss here. The first one is lipase, and this works with bile to break down fat like previously discussed. There's also proteases, and these break down proteins. And finally, there are amylases, and these break down starches into sugars. All right, nextly, the exocrine pancreas also secretes sodium bicarbonate, and this has three major functions. The first one is it serves as a buffer to acidic gastric juice and chyme. It inactivates pepsin in the stomach, and it also provides an environment for digestive enzymes to thrive because they operate in an optimal pH range, and this makes sure that the pH doesn't get too low. So we'll see in this figure over here that firstly, carbon dioxide enters the pancreatic cells from the interstitial fluid, and this reacts with water in order to create bicarbonate and also a proton, which creates the acidity in these cells. And this, right next to the 1 here, it says Ca, and this represents carbonic anhydrase. In order for this reaction to work, it uses carbonic anhydrase, which is the reason that these cells require very large amounts of it in order to create bicarbonate. And then at 2 here, you can see that chlorine enters the cell from the interstitial fluid, and then it actually exits the cell into the lumen of the pancreas, and then it eventually re-enters the cell in exchange for bicarbonate to exit the cell. So that's kind of the driving force to get bicarbonate to enter the lumen of the pancreas. And then finally, number 3 on this diagram shows how the transfer of sodium and bicarbonate actually create an osmotic gradient. So water falls and creates the watery portion of the sodium bicarbonate solution. And it's also important to note that small amounts of bicarbonate are also secreted by the duodenal cells, but most is produced in the pancreas. Next, we'll talk about the endocrine pancreas secretions. And these, firstly, are stimulated by the hormones in the parasympathetic nervous system via the vagus nerve. So the first one is cholecystokinin, or CCK, and this stimulates acinar cells to secrete enzyme-rich pancreatic juice and stimulates the secretion of secretin. And secretin causes duct cells to release the bicarbonate-rich pancreatic juice. And then there are 4 main endocrine secretions that we'll be talking about. The first one the first three actually are secreted by the islets of Langerhans, and you can see that here on the figure on the right. It's actually the lightly stained region in the cross-section of the pancreas. So the first secretion that we're concerned about is insulin, and we've talked about that a lot in previous lectures, but this regulates the level of sugar in the blood, and they're produced by beta cells. The second one is glucagon, and this releases stored glucose or glycogen from the liver, and it favors insulin release, and eventually it inhibits glucagon release in a negative feedback. The third one that's secreted by the islets of Langerhans is amylin, and this is co-secreted with insulin, and it's important because it helps control appetite, and it also delays stomach emptying. And then lastly, gastrin is, it stimulates the stomach to make gastric acid, and it's mostly made by G cells in the stomach, but some are also made in the pancreas. And finally, we'll talk about some common pancreatic digestion problems. The first one that we'll be discussing is diabetes, and we've talked about this a lot previously, but we'll mention a little bit here. Type 1, as we've discussed, is an autoimmune disorder where beta cells are destroyed and don't produce insulin, and type 2 produces some insulin but not enough. And this is important in regards to the digestive system because it can actually cause gastroparesis, which is a reduction in the motor function of the digestive system. Secondly, we'll talk about pancreatitis. This can be an acute or chronic issue, and it's inflammation of the pancreas. And it's where digestive enzymes attack the pancreas and cause severe abdominal pain, and it affects digestion because enzymes aren't available, and it leads to diarrhea, weight loss, and malnutrition. Finally, the textbook actually mentions cystic fibrosis, so we'll also go over it a little here. So cystic fibrosis is a genetic mutation, and it causes the secretion of chloride and fluid to cease, but the goblet cells continue to secrete mucus. This results in thickened mucus in the pancreas and also in other areas such as the lungs and some other organ systems in the body. But in regards to the digestive system, the thickened mucus clogs the small pancreatic ducts and prevents digestive enzymes from entering the small intestine, which can have similar effects of pancreatitis, albeit they are caused from different reasons. And then that finishes our lecture.